And now let us move on to codons and anticodons. Codons, which are found in mRNA, are the complementary triplets of the code triplets of DNA genes, while anticodons are found in tRNA that is complementary to codon. These two terms are related to each other. Codon is a 3-nucleotide stretch of sequence which codes for proteins. Looking back at the central dogma of biology, this begins with a DNA molecule. From the DNA, it produces mRNA, and from mRNA, it produces protein. mRNA, which have different bases coming out because it is a single-stranded structure, and 3 of those bases coming out are called codons, which are specific for coding a particular amino acid. In mRNA, there are many types of codons, which is placed in a chart called genetic code. When you look at the genetic code, you will find many different codons as well as all different amino acids that those codons are coding for. Codon means 3 nucleotide in which each of the 3 nucleotide is specific for coding a particular amino acid. Now, to interact with those 3 codons, we need opposite 3 nucleotide sequence which will sit onto the codon and read that codon, then bring out the right amino acid. Because during the protein synthesis, we require 3 major molecules. One is the mRNA, which will be coded into proteins. To make it more simple, we have this strand of DNA that will move from 5 to 3. So for the complementary strand, you will have A paired with T, T paired with A, and G paired with C, and so on and so forth. Messenger RNA is similar to DNA, but instead of thymine, you have uracil. So now for this complementary strand, the DNA will split into 2 by the polymerase enzyme. Then there will be single DNA strand that is left, and eventually, the T of single strand will pair with the A, and A with the U, and C with the G, and so on and so forth. Now, messenger RNA pairs with the transfer RNA. The messenger RNA will be the codon, and the transport RNA will be the anticodon. The transfer RNA has uracil, therefore A will pair with U will pair with A, and G with C, and then so on. Now, let's find the amino acid by using an mRNA chart, so I will use the messenger RNA codon. mRNA will code in triplets. AUG, which is a start codon, pairs up with methionine. Then I have CCU, and it pairs up with proline. Then CUA pairs up with leucine. And lastly, GUA pairs up with valine. These 4 are the amino acids.